everyone, welcome to this explainer. Today we're diving straight into the fundamental unit of life, the cell. We're going to break down the exact data, structural logic, and clear definitions straight from our source material. And, keeping with our specific focus for today, we're doing this entirely by building a mental blueprint. No distracting diagrams from the text, just pure text-based logic and structural breakdowns. Let's get right to it.
So, here's our roadmap for today. We'll start with the basics of life, move on to cell shapes and functions, then tackle boundaries and fluid mosaics. After that, we'll look at the cell wall, head inside the organelle factory, and finally, wrap things up with the ultimate cell quiz to test what you've learned.
All right, part one, the basics of life and the foundation of cells. Let's jump right into a classic comparison. Plant cells are essentially built for tough, rigid structure and totally autonomous energy production. They've got a dense cell wall, chloroplasts for making food, and a massive central vacuole to keep their internal pressure high. Animal cells? Well, they strip away that rigid wall in the chloroplasts. They opt for much smaller vacuoles, but they uniquely pack a centrosome to help out with cell division. It's honestly a brilliant biological trade-off between structural rigidity and flexibility, you know? Now, whether we're talking plant or animal, inside both of these cells, you're going to find the cytoplasm. Think of it as a half-liquid, jelly-like matrix. But look, a cell isn't just a wobbly blob of jelly, right? It needs actual structural integrity, and that's exactly where the cytoskeleton comes into play. It acts as this absolutely crucial physical framework that supports the cell and maintains its specific functional shape. Moving right along to section 2. Cell shapes and functions, where form absolutely dictates function. So a cell's physical structure is always tied to its biological purpose. Just think about your own body for a second. Mammalian red blood cells, or erythrocytes, are round, biconcave discs. And get this, they famously lack a nucleus in adults just to maximize how much oxygen they can carry. On the flip side, your white blood cells, the leukocytes, have an amoeboid, totally irregular shape because they're basically the soldiers of your body, squeezing through tight tissues to hunt down invaders. Contrast both of those with nerve cells, which are super long and branched, making them perfectly specialized for transmitting electrical signals across huge distances. Like I said, form truly equals function here. Okay, section 3, let's zoom in on boundaries and fluid mosaics, specifically looking at the cell membrane. Every single cell requires a protective barrier, and that boundary is made of a lipid bilayer. It's kind of like a fascinating chemical sandwich. The outer components are these hydrophilic, water-loving heads that are polar and contain phosphate. Hiding away safely on the inside of this membrane sandwich are the hydrophobic, water-hating lipid tails that have no net charge. This highly specific chemical arrangement naturally forms an incredibly stable barrier in a watery environment. To really wrap our heads around how this membrane behaves in real time, we use the fluid mosaic model. This was proposed by Singer and Nicholson back in 1972, and it totally shifted the paradigm. Instead of a rigid, static wall, they described the membrane as this thin, selectively permeable, living layer. Just picture massive membrane proteins acting like giant icebergs, right? Just constantly drifting and floating around within a vast, fluid sea of lipids. It's a pretty wild mental image, but it's incredibly accurate. Next up, section 4. We're looking beyond that plasma membrane to a much more rigid boundary. The cell wall. Now, not all cell walls are created equal. In fact, there's incredible chemical diversity across the different kingdoms of life. Plants, as you might know, rely primarily on cellulose, bolstered by things like hemicellulose, pectin, and proteins. Fungi, on the other hand, go a totally different route. They build their walls out of chitin, along with galactins and mannins. Meanwhile, bacteria depend on a completely different substance called peptidoglycan, which is also known as murine. So what we're seeing is nature finding 3 distinctly different chemical solutions to the exact same structural problem. If we focus just on plants for a second, their cell wall actually isn't static. It physically transforms as the plant ages. First, neighboring cells are held together by the middle lamella, which essentially acts as a biological glue. Interestingly enough, as a plant matures, its initial outer layer, the primary cell wall, actually starts to diminish. In its place, a thick, super tough secondary cell wall packed with lignin is added to the inner side. This is exactly what gives older plant tissues, like wood, their extreme rigid strength.
All right, let's step back inside the cell. Section 5. Inside the organelle factory.
To really master cellular anatomy without relying on pictures, it helps to organize all these tiny cellular machines into 3 clear mental buckets based on their membranes. First, we have the heavily fortified double membrane-bound organelles. Then, the single membrane-bound ones. And finally, the structures that are completely membrane-free. Looking closely at the heavy hitters, the ones with double membranes, we find the absolute most critical infrastructure of the cell. You've got the nucleus, acting as the ultimate control center and safeguarding your chromatin. You have the mitochondria, universally famous as the powerhouse of the cell, generating energy across its internal crista. And in plants, you've got the chloroplast, the dedicated site of photosynthesis packed with grana and stroma. These three are just so vital that they require a double layer of lipid protection. Meanwhile, the rest of the organelles are basically handling the day-to-day logistics. The single membrane organelles are all about manufacturing and waste. The endoplasmic reticulum, coming in rough and smooth variations, acts as a manufacturing network. The Golgi apparatus handles shipping from its cis-face and packaging from its trans-face. Then you have the lysosomes, holding digestive enzymes and fiercely nicknamed the suicide bags of the cell. Finally, in our completely non-membrane-bound category, we have the ribosomes, the fundamental, everyday workers responsible for literally all protein synthesis.
Okay, Section 6, the ultimate cell quiz. Let's lock this in and see what you remember.
Question 1. Think back to our section on cellular boundaries. Who were the two scientists that brilliantly described membrane proteins as icebergs drifting in a fluid lipid sea? Do you remember their names?
Yup, the correct answer is Singer and Nicholson, who published this absolutely foundational model back in 1972.
All right, let's move to question 2. Which of the following structures is so fundamentally essential for life that exists in both simple prokaryotic bacteria and our own complex eukaryotic cells? Is it mitochondria, the endoplasmic reticulum, ribosomes, or lysosomes? If you guessed ribosomes, you hit the nail on the head. Because they lack a membrane entirely, they're present across all domains of life. Though, as you can see here, they do differ slightly in size, sitting at 70S in prokaryotes and a slightly larger 80S in eukaryotes. Moving on to our final multiple-choice question. What specific structural component in a simple eubacteria actually bears a striking resemblance to our own complex eukaryotic cells? Is it the plasma membrane, the cell wall, the nucleoid, or the plasmid? The answer is the plasma membrane, which is super cool because it proves that while different kingdoms differ wildly in their cell walls, using things like cellulose versus peptidoglycan, the basic fluid mosaic lipid blueprint of the plasma membrane is remarkably universal across all of life. So we'll leave you today with this final provocative thought. Considering how incredibly strictly a cell compartmentalizes its factory floor, separating precious DNA behind a double membrane or locking acidic digestive enzymes safely inside single membrane lysosomes, how quickly would life just completely unravel if all those protective internal membranes suddenly vanished? It really makes you appreciate the highly structured, perfectly partitioned reality of the microscopic world. Thanks for tuning into this explainer, and keep exploring.